Okay, now it's also live on the YouTube. Okay.
So PPT I have to share from my side, or you will share it from your end?
You can share from your side. That will be easier.
Okay, fine.
Just in case you keep it ready from this side as well, you can send it by email also, so that we would have a copy.
Already sent by email on both the email IDs you have shared.
Then perfect.
You can check. I have shared it already. Please check.
Yeah, I will download it. Thank you.
मेरी स्लाइड दिख रही है? So how do I share this? If Pushpin, how do I share the slides in this?
In the share screen, you see there is a one green.
Okay, share screen. Fine. Just मैंने भी सोचा था ऑफिस जाने का. स्क्रीन क्यों नहीं शेयर हो रही?
Memory will share screen. पहले वो पावर पॉइंट ओपन कर रखी है आपने?
मैंने ओपन कर लिया है.
Okay. Then you have to identify that particular open file from. If you open it on desktop, or
I have opened it on desktop.
okay, and
Let
then
me close everything else.
Yes, from on the desktop, during the share screen option, you have to identify that particular file and put it in the.
Just a second. Zoom has an option for share screen. Okay.
Yes, it's in the. It's.
Okay, fine, got it.
The green one.
Just a minute.
Yes, it's perfect. Okay. The participants are starting to join. I think we can begin with small round of
shall we begin,
just maybe in
or we can wait for some more participants to join? It is
yes,
still 2 minutes to go.
actually we can introduce ourselves. Just want to welcome all the participants and ensure that they are not having any difficulty. So just one request to all of you: if you have any question, please use the button raise hand, and if you have any question, you can type in the chat. So I request Divya Chauhan and I hope the problem with the echo sound is no longer there.
I think it's fine now. Is it fine?
Yes, but just please keep your slides in the slideshow mode, please.
Pardon, please.
Can you put your slides in the? Can you put your presentation in slideshow mode from the desktop?
Fine.
From wherever you have kept your slides open. Yes, now it is in the slideshow mode, and with that we can begin also. And it's actually being live streamed and it is being recorded also. So just for information of all the participants. And it's my pleasure to welcome Dr. Gauri Mishra, who I have the pleasure of knowing since the time of my master's actually, when I was doing my master's degree. I request other participants to keep themselves muted, please. Bridge and Anisha, can you keep yourself muted for the time being? If you have any question, you can put that in the chat box. Thank you. Otherwise, I would have to mute all. So this is a great pleasure for me to host this talk. Today's speaker is Dr. Gauri Mishra, who is a scientist grade 2 at National Institute of Biologicals at Noida. She has more than 15 years of experience in molecular research and structural biologist. There is some echo sounds from some place. Let me figure out. So listen to the sound again. Introduction for now. Inviting Dr. Gauri Mishra to start her presentation.
Just a second, there is some echo sound. I'm trying to figure it out, how to remove.
Okay, now I think is it better now?
I think, is it better? Can you hear me now clearly without the echo sound?
Yes, we can hear you. So Dr. Gauri Mishra has done her PhD from CDRI Lucknow, and after that she has done her postdoc work from Canada. Has several research publications and has also published or edited 5 international books from highly reputed publication houses, and she's running funded research projects from several funding agencies. She's also heading the laboratory for COVID kit quality control, and she's also heading the molecular diagnostic laboratory at National Institute of Biological. With that very short introduction, I invite Dr. Mishra to deliver her talk. Floor is yours, Dr. Gauri.
Thank you so much, Dr. Pushpendra. It's my pleasure. First of all, I thank Dr. Das, Director of the NIRTH. I also thank my director, Dr. Anup Avnikar, and then I thank Dr. Pushpendra for giving me this opportunity to share my research-based experience, my present experience with all the participants who have joined. So good evening, everyone. Welcome all the participants. I hope it will be a pleasure to interact with you virtually, and hope you will have some gainful insights through this presentation. So when I take ahead with this, I would like to share that the initial few slides, since the primary audience is our students from undergrads and postgraduates, we have a diverse audience from the students to the research scientists. So there will be few slides which will be basically having the basic knowledge, and then I will have few examples from my own research work. Okay, so basically my presentation is focused on the structure-based approaches for drug discovery. So in one slide, I would like to introduce you to my institute. I am presently located at National Institute of Biologicals, Noida, where I am, as Pushpendra has just briefed, I am heading the molecular diagnostic and COVID kit lab. And we have diagnostic, therapeutics, vaccine and support labs in our institute. Most of the labs are NABL accredited and CDL accredited. The best part is that this is the only institute that provides hands-on training to the most sophisticated infrastructure experimentation like capillary electrophoresis, SPLC, etc. So all the student audiences, whenever they intend to do a short-term research work or a 6 months research project, they can always explore the National Institute of Biologicals Noida website, and they will always find an opportunity to get a very good state-of-the-art infrastructure training, and that to a practical hands-on training. So in our institute, we are actually performing the quality control testing of around 277 biologicals. So that is a brief about my institute, and it is the only institute in the entire Southeast Asia which is involved in the quality control regulation of biologicals. So I'm coming now to the basic theme of this lecture series, which is genomics in drug discovery. So genomics is basically falls under the domain of the genetics, And the main areas which are involved is mapping, sequencing, and after that it is related to the functional analysis part. So genomics, there is a difference between a genomics and a genetics. Genomics involves the study of all the genes at the DNA, mRNA, and proteome level, as well as the cellular and tissue level. I think students are familiar from their school days with the central dogma of life. DNA replicating itself to DNA, then after transcription forming the mRNA, which then codes for the proteins, which is a translation. So at every level, when you say genetics, generally it shows that it is a single gene, one at a time. However, genomics gives you a global picture where it provides the opportunity that you can examine all the genes in the entire system, or you can say if you have a human being, you can examine all the genes that are present in a particular human being or an organism. If we go to the, I will just in a few seconds recapitulate the history of this. Genomics was first developed by the Fred Sanger in early 1970s. In 1972, Walter Fierce and his research group became the first research group to sequence a gene, where they have done the sequencing of a bacteriophage gene MS2. In 1995, thereafter, Hamilton Smith and his team became the first to sequence the entire genome of the Haemophilus influenzae. From there came the concept of the sequencing of the genomes. So basically, how does sequencing of the genomes give an advantage to the current boom in the data? So sequencing the genomes helps everyone to understand how the genome as a whole and how the gene at an individual level helps and direct the various developmental processes, growth and maintenance of an entire organism. And there is a very close relation. See, it is like if you know the A, B, C, D, E, F, G, then only you can join the letters, you can make the sentences, and thereafter you can write a entire English essay. It is simply, although you have 4 nucleotides, you have adenine, thymine, guanine, and cytosine, but different combinations of this ATGC is basically responsible for different genetic compositions. And when you sequence the whole genome, you are able to get insights about all the possibilities. How does an organism grow, how the development takes place, how the maintenance of the entire organism is taking place. Not only this, the genome sequence is actually be able to tell you that, okay, if there is a difference in a single gene, how the difference or a variation, as we say in science, at a single position in a gene can lead to some disease, or how a single variation at a particular position will affect the phenotype of a person. This is how the sequencing of the genomes helped during the course of its evolution. Now, the basic methods through which this genomics steps involves is the construction. First of all, there is a construction of genomic and cDNA libraries. Then you have DNA hybridization, where you require small probes which are complementary to the gene sequences. This DNA hybridization is then followed by the restriction enzyme mapping. DNA sequencing, you can sequence the entire DNA, or you can just do the specific gene sequencing followed by the PCR amplification. Most of the students must be aware of the classic sequencing methods that they would have studied in their coursework: the Sanger chain termination method and Maxim Gilbert method, which is basically a chemical sequencing method. However, there are advanced sequencing techniques that are routinely used, which you must be familiar of with the name at least. We say next generation sequencing methods. So as Dr. Pushpen also earlier has explained in detail about the applications and the details of the NGS, so I will just go through quickly. NGS involves, there are different next generation sequencing methods. There is 454 pyro sequencing. You have iron torrent semiconductor sequencing, sequencing by ligation, reversible terminator sequencing. Illumina is a very well exploited platform nowadays. And then nanopore sequencing, but certainly nanopore sequencing is a very, very costly method. The principle behind the next generation sequencing is similar to what is the Sanger sequencing, which relies on the capillary electrophoresis. Here, the genomic strands are fragmented, and each fragment is identified by the emitted signals when the fragments are ligated against a template strand. This NGS method uses an array of sequencing methods that actually combines the different techniques developed in Sanger sequencing, so that you have millions of reactions running in parallel, resulting into a high throughput output. You can have an entire sequencing done in, say, a very fraction of time. So here you can see the diagrammatic representation. You have a genomic DNA, which is subjected to either sonication or the digestive restrictive enzymes, followed by the this DNA fragments are then ligated to the adapter sequences. These fragments are generally attracted to one another by mostly the hydrogen bonding, and once the ligation of two fragments occurs, these fragments modified with the adapters as the 5-hydroxy terminus, they will not dimerize against its with each other. As a result, this you can actually read the sequences of these fragments, which are later on mapped, and then once the mapping is done, they are compared with some template sequences. Coming to the, as I have just explained, genetics is the broad domain under which genomics is there. Then we have different subdivisions of genomics. We have structural genomics, functional genomics, and a comparative genomics. When I talk about structural genomics, that has been a forte since my doctoral days. Structural genomics means you have an entire genome which is, say, codes and translates into proteins. So if you have a structure genome coding for translating into some functional protein, so structural genomics involves an understanding or determination of the three-dimensional structure of all the possible proteins that are coded by a particular genome. So if you have a knowledge of some genome, and you gather an information using various genomic approaches about the possible three-dimensional structures of all the proteins that are encoded by that genome, is what does the structural genomics constitutes. Now, from the structure part, we then move forward to the functional genomics. Now, you know, okay, this is the genome coding for these proteins. This is the basic structure of these proteins. Again, it is not necessary that all the proteins that are coded by a genome are functional. There will be some proteins that may be non-functional and some proteins that will be functional. So here, all those proteins that are functional, those proteins that are coded by a genome, what is their functional? This correlation is established by the functional genomic methods. Again, based on the information that we gather through structural genomics and functional genomics, we have an advantage to perform the comparative genomics. Here in the comparative genomics, say if you have an information from a human organism, from the human genome, you can simultaneously compare the information gathered from the human genome to the mouse genome, to the cow genome, or any viral genome or any bacterial genome. So comparative genomics basically makes use of all the above two subfields, where you have an information for the structure of the proteins that are coded by a certain genome. Then you decide under functional genomics what are the functional proteins that are coded by this structural genomics, and then you can have a fair idea about the comparative genomics. Moving ahead, as I have just briefed, so you have this central dogma, DNA replicates, it transcribes to RNA. When in case of cDNA formation, there is a reverse transcription where the cDNA is formed. Once it translates into a protein, which is a three-dimensional compact structure. To recapitulate the basic principles of protein structure, because genomics, I have been mentioning this again and again, genome coding for proteins. This is specifically meant for for the student audience. So you have a basic principle is protein structure. You have a primary structure where it is like a string of the alphabets. Primary structure is basically the combination of the amino acids. Now this linear sequence of the amino acids is your primary structure, which you can determine using the structural genomic parts. This primary structure then codes for a secondary structure. As you can see, alpha helix is kind of a ribbon-like structure. Then you have a beta sheet. This beta sheet is in the form of a arrow. This primary structure folding onto itself to form a alpha helix and a beta sheet structure. This is your secondary structure. These secondary structure elements, alpha helix, beta sheets, then you have loops, coils, they combine and they interact with each other, undergo the protein folding to form a tertiary structure. Now you can say a protein can have a single chain or protein can have a multiple chain. If a protein has a single chain, for example myoglobin, you say the highest after folding it will only form the tertiary structure. However, if you have a protein which has more than one chain, then obviously protein having more than one chain and more than one subunit, they will form the quaternary structure. For example, you have in hemoglobin. In hemoglobin, you have two alpha chains, two beta chains. So if you have a protein that has only single peptide chain, the highest structure that it can gain is the tertiary structure. However, the protein which has more than one polypeptide chain, it can fold up to the quaternary structure. Now, coming to the application part. So you have a gene coding for a protein. Now, these genomics sequences actually guide primary sequence, as I have just mentioned, primary structure, the string of the amino acids. So there are only 22 amino acids. Now in what combinations these 22 amino acids, they only undergo folding in million and trillions of ways to form millions and trillions of proteins. Although you have only 4 nucleotide bases, A, T, G, C, and you have only 22 amino acids, but a different altogether combination. These amino acids undergo different ways of protein folding to form millions and trillions of proteins. But what kind of primary structure will be there, what kind of secondary structure each protein will form is always unique. And this uniqueness is guided by the genome sequence. So from the genome sequence, it is determined which protein will form which structure and what function that protein is going to perform. However, the actual players in the cell are the proteins. So these are the protein, as you know, are the machineries that help in the communication, cell signaling, growth or death of an organism. When we relate to this gene, protein to disease, then each protein has particular structure. Because of that structure, it has a particular function. Structure is coming from the gene part. So now most of the proteins that are involved in different diseases, or we say they are the drug targets. Any protein which you can target for this drug discovery. Suppose there is a protein which is functional in a normal organism. If it has some mutation or if there are changes in the genome sequences, even if there are at times point changes, for example, sickle cell anemia, Just mutation of a single amino acid results in a entire shape of the red RBCs. So in sickle cell anemia, just a single mutation in an amino acid sequence completely changes the structure of the RBC. So that point mutation is responsible for changing the RBC into a leading to a sickle cell anemia. So most of the proteins, if they are functioning in a regulated fashion, they are actually the building blocks of an organism. However, if a protein loses its function, then there are possibilities that it will lead to disease. This we call as dysregulation of functioning. Now that protein we say is a drug target, means if you know the structure of that protein, you can design a drug which can actually go and bind to that protein, and the protein that has dysregulated due to the binding of that molecule will stop functioning. Thus, it is a drug target which can be regulated by designing a novel molecule. This is from where the concept of structure-based drug design has come. So drugs generally work by binding specifically to a particular protein. So as I have mentioned, this concept of structure-based drug design is a very, very important concept in present scenario for drug discovery. Initially, if you say drug discovery, generally from the bench to the market, to the clinical trials, it actually takes around 12 to 15 years for a drug to be designed for a particular disease. But when we say about the structure-based drug design, it is a very rapidly growing area because you have a genome, you have a knowledge from the genome, you have understood the composition of the genome using structural genomics. From the structural genomics, now you have used the functional genomics and you know that a X genome is coding for so many functional proteins. Once you have the functional proteins, you can use the comparative genomics, and then from the comparative genomics, you can actually narrow down what are the drug targets. Drug targets, as I have just mentioned in the previous slide, are the proteins. Once you know those drug targets, the structure of those drug targets, you can design drug targets are like, you can say proteins are the three-dimensional structures. You can design a molecule that goes and binds to that particular protein in a complementary manner so that any protein which is dysregulated stops its functioning. So this structure-based drug design has led to a different drugs that are currently used in the market. The determination of now the question comes, how do you determine the three-dimensional structure? You have a tertiary or the quaternary structure. The main methods that are used for determining the three-dimensional structure of any drug target is the X-ray crystallography or NMR. In a coming slide, I will just brief what are the generally this structure-based drug design, what is the workflow, how this structure-based drug design is performed. So for this, you generally choose drug target, which is generally a protein that is very crucial for an organism, but somehow due to some genetic mutation or some environmental effect, the protein has lost its function. So based on that, you have chosen a drug target, which is a protein. Now, you obtain pure preparation of target in solution, means you purify that protein, right? And once you purify that protein, you determine the three-dimensional structure of that protein by either crystallography or NMR. Now, once you have determined the structure by crystallography or NMR, there are two methods. Either you can use the in silico methods computationally also. There are two methods to determine the three-dimensional structure. One is the computational method and another one is the experimental method. Experimental method for protein structure determination is crystallography or NMR, whereas computational method involves homology modeling, where you are familiar with the similar structures and you modify your sequence and you align your sequence with the structures that are known. So this is the computational method. In any case, either you are using the computational method or you are using the experimental method. Once you know the structure of the drug target, which is a protein, you can actually analyze that structure to design the inhibitor. You can determine which are the sites on that protein where any molecule can bind and what will be the effect of that molecule on that protein. For this, you must have heard of the term, we do the molecular docking. So generally, we do not this inhibitor binding to the protein molecule. It is not generally done in one or two methods. It is generally we take millions and hundreds of molecules that are docked against that drug target. And once we dock several molecules against the drug target, then we analyze, okay, what are the binding energy and which is the best drug target. How do we decide which is the best molecule that is binding to the drug target? Based on the binding energy. Those compounds which has the highest negative binding energy, they are we propose that they are the best inhibitors against those drug targets. We analyze and rank the compounds based on their binding energy and selectivity. Suppose it is not good, we again go and reiterate, we modify we call this, see, right now we are just screening the compounds. Right now they are not inhibitors. At this stage we call them leads because they are not yet established experimentally that they are the inhibitors. So they are the lead compounds. And if they do not show very good binding energy, you can actually at this stage using computational methods modify these compounds, optimize these compounds and again go for the docking, and then again you check their inhibition binding capacity against the drug target. These compounds, once you know this is the best compound, you can either, if they are available commercially, you can purchase them or you can get them synthesized. Till now, I again repeat, these are not the inhibitors, they are still the leads. Now, these lead molecules, they will go and bind to the drug target again. Initially, you determine the three-dimensional structure using NMR. Now also, once the compounds bind to the particular protein, you can actually determine the compounds in combination with the drug target using NMR or crystallography. We call it as the co-crystal structure or combined structure in the NMR solution. Once you know the structure, then from that you can do the kinetic studies. You can determine their binding inhibition, binding affinity, inhibition affinity, and then you can actually, these compounds, once you know these are the leads which are effective against the drug target, these compounds are then tested for their potency. Then they undergo different clinical trials, phase 1, 2, 3, and 4. And once they pass the clinical trials, then you call these lead compounds as drugs. So this is an entire snapshot or the flow, how right from the structure genomics to functional genomics, how this structure-based drug design actually directs the drug discovery procedure. Now coming to the second part, that was some of the basics I thought it would be beneficial for our general audience. Now I will just give one or two specific examples from my own research work. One of the structure-based drug design inhibitors we have screened was against the Bacillus anthracis protein, that was nucleoside diphosphate kinase. You can see a beautiful structure over here. NDK is nucleoside diphosphate kinase. The source was Bacillus anthracis. As you can see, a beautiful structure. This protein is a trimer, means it has 3 chains, and actually it is a dimer of trimers, means 3 chains on top and 3 chains on bottom, so it is a dimer of trimer. This beautiful crystal structure, as I have told you, nucleoside diphosphate kinase is a protein from Bacillus anthracis, which is a drug target, very important drug target for in Bacillus anthracis. We determined this 3-dimensional structure using X-ray crystallography. Not only this, under the domain of the structure-based drug design, we have also done this was the structure we determined using the experimental method, X-ray crystallography. We also had the structure of a mutant for this protein, and we called it as the G19B mutant, because at the 19th position of the polypeptide chain, glycine was replaced by valine. Somehow when we mutated this glycine to valine at the 19th position, the protein did not form a crystal. So what we did, rather than going for the experimental method of crystallization, we opted for a in silico computational method to determine the three-dimensional structure of glycine 19 valine mutant. And the best part was that then the structure that we determined for the wild type NDK, which I just showed, this structure and the structure of the glycine 19 valine mutant, we overlapped on to each other to understand how this mutation affected the function of this protein. See, since it is a nucleoside diphosphate kinase, obviously kinase means it will add to the it will add phosphate group to that particular phosphate will be it will be a phosphate adding enzyme. So once this mutation was there, then we did lot of structural analysis, and we realized, if you can see, there is this loop, rigidified loop. In the wild type structure, this loop, as you can see in the blue color, was very flexible. Once this glycine 19 valine mutation took place, if you see this green color, glycine 19 valine mutation is very far, if you say it is very far from that loop. But you can imagine, when a structure folds onto itself, a distant mutation could actually lead to the rigidification of this loop. So the wild type structure, the loop was very flexible. But when this glycine at the 19th position was replaced by valine, this loop got rigidified. So what happened? After the rigidification of the loop, we again tested for the nucleoside diphosphate kinase activity of this protein, and we realized this protein was no more phosphorylated. Its phosphorylation capacity decreased almost 300 times. So you can imagine how the structure of a protein is related to function. Under the normal structure, protein was capable, since it is a kinase, it was capable of phosphorylation. But when a mutation was incorporated into its peptide chain, then automatically this loop became rigidified. This rigidification of the loop affected the function of this protein, means its capacity to phosphorylate the substrates reduced. So this we actually published long back. But however, this is a beautiful example I always wanted to share with the students, that how a structural protein structure, understanding of a structure of a three-dimensional structure of a protein can actually help you in understanding the changes in the function of a protein. This was the beautiful crystal that time we got for this NDK. We got this highly purified protein, and this was the diffraction data that we received. Now I'm going to introduce to a very recent study in our group, which we published in Nature Scientific Reports in 2018. Again, here we had a very important protein, microtubule assisted serine threonine kinase. This serine threonine kinase is a very important drug target. In my initial slides, when I was introducing you about the drug targets, I mentioned that drug targets are usually the proteins that play a very crucial role in normal organism. Sometimes they get dysregulated, then you have to design a way to inhibit their overexpression so that this dysregulation is controlled to prevent the disease progression. Now, mast cell is an important protein microtubule assisted serine threonine kinase. This serine threonine kinase is involved in the cell cycle regulation. I think most of the students are aware of the general cell cycle. You have a G1 phase, you have a synthetic phase, you have a mitotic phase, and you have a G2 phase followed by interphase and again a G1 phase. So you have a cell cycle progression. This kinase is involved in the regulation of transition from G to M phase, G2 to mitotic phase. So, and this kinase is very important. Its overexpression is related to in different types of cancers: breast cancer, lung cancer, head and neck squamous cell cancer. Under normal situations, this is involved in cell cycle regulation. But under cancerous conditions, this protein gets overexpressed and its regulation, it gets dysregulated. Its normal function of G2M regulation is hindered. Now we were interested, since this is a very important now, therefore it is an established drug target, an important protein, which if you stop the overexpression, then you can somewhere stop the progression of that particular type of cancer. Since this protein was involved in different cancer types, so what we thought to start with the computational work, because somewhere that is the best part of the computational work at the initial stage is that it saves the time and the resources. So what we did, we took the three-dimensional structure of the master from the PDB, protein data bank. We screened lot of databases. One was zinc, another was a natural compound database. Several hundreds of compounds were screened against this protein. Compounds in the initial stage underwent ligand preparation. Protein structure also, three-dimensional structure, we did the protein preparation. Then we did the molecular docking. After molecular docking, we did the simulation studies. And after simulation studies, molecular dynamics simulations, we did lot of structural analysis part. And this structural analysis part actually gave us this. And what is this? We could actually identify new inhibitors against this mast protein. This another nickname for this mast protein is human great wall kinase, a very important drug target in different cancers. Again, I am repeating, regulating the cell cycle phase from G to M. Now, this was one interesting finding in our studies, where we could actually identify 10 inhibitors, 5 from the natural source and 5 from the synthetic source. And this was all computational work. Now we wanted to be sure that the compounds that we have identified, they are actually inhibiting, or they are just the false positives that we are getting. So to validate our in silico findings, we moved ahead with the molecular dynamics simulations, and we could understand that, as you can see, this was the three-dimensional structure of this protein, and you can see these compounds beautifully interacting with the active site. You can see there are hydrogen bond interactions, and there are multiple hydrogen bond, and even as you can see with the aspartate and the glutamate, there were ionic interactions. So these ligands, these compounds, which we still call as the leads, they were given by the computational network, computational studies. We could actually find till now, this is an important drug target, but no group has reported any inhibitor against this protein. This was the first study where we could actually report significant inhibitors against this protein from the natural as well as the synthetic sources. Now, presently, although I have not shared that data here because we have a lot of data, what we did, but we are about to publish that these compounds, now we have done the cell-based studies. We have done the experimental validation in the breast cancer cell line. And we could actually find that these compounds are showing very good IC50 inhibitor concentration at which 50% of the breast cancer cells die. They are showing high specificity against this protein. So these compounds actually are showing good inhibition in our breast cancer cell-based studies. Not only this, we have also now given our compound-based, the cells, breast cancer cells that are treated with these compounds, we have subjected them, we have outsourced them for the next generation sequencing. And from the NGS data, now you can imagine, see, it is all complementary approaches to address one biological question: how to regulate a dysregulated drug target, that is mast cell. So what we did, we started from the in silico work. We identified the compounds that could actually inhibit this protein. We have experimentally validated these compounds in cell-based studies, and now using the comparative genomics with the wild type untreated protein, we have now subjected these cells that have been treated with these new inhibitors, and we have subjected them to the next generation sequencing. This next generation sequencing will provide us the insights that when you are treating a protein with X inhibitor, which are the functional pathways that are disrupted, which are the functional pathways, which are the gene partners that are interacting with the mast cell. So all this insights we will get from the next generation sequencing data that we are going to receive. So this is an integrated approach, from the in silico studies to the cell-based studies to the genomic studies involving the next generation sequencing. There is a second study that we have also undertaken recently. This study has been recently published in Briefing in Bioinformatics with an impact factor of 11.6. Again, TAD. This TAD is a transcription factor. Right? As I have just told you, the previous protein is a mast cell, is a serine threonine kinase present in the nucleus. TAD is a transcription factor. This is a second drug target, which is very crucial for the therapeutic intervention of cancer. It has been reported that TAD binds to there are 4 types of TAD in the cell. 1, 2, 3, 4. It is reported that TAD2 binds to the palmitic acid, and once it binds to the palmitic acid, it leads to the transcriptional coactivation. Now we have identified an inhibitor that has high affinity to this protein, TAD2, transcription factor. Now once this inhibitor binds to the active site, 3-dimensional active site of this protein, this palmitic acid is able to bind to the active site. As a result, the transcriptional repression takes place. TAD2 is unable to perform the transcription. We have identified the new inhibitors again using in silico approaches. As I have mentioned in my previous flow chart, we have did the molecular docking studies, followed, and the results that we have got, the lead compounds that we identified using the docking studies from the computational approaches, we have validated those compounds, their interactions with the TAD2 protein, which is a drug target, using molecular simulation studies. And this is some of the data. For students, it may be slightly complex. Let me give a try to make it easier for you to understand. So this, as you can see on the x-axis, is the simulation time in nanosecond. The data, once you have a drug target, which is a TAD2, we screened against the databases, we identified the inhibitors that docked against the drug target. We could identify the initial lead compounds which exhibited a high binding energy. Once we knew those compounds, we subjected them to a 100 nanosecond simulation. 100 nanosecond simulation is quite a good time to rule out that these are the actual binders that bind to the drug target and they are not the false positive, false negative. So as you can see, there is a smooth line. You can see that with time, the entropy of the system is also getting stabilized. This shows the root mean square deviation is also not very high. The configurational entropy of the ligand complexes, 4 inhibitors binding to the drug target, is also getting stabilized. This actually is a validation of our in silico results that we got from the docking studies, that there is some possibility that these compounds, if they are experimentally validated, they will be good leads that can be carried forward for cell-based studies. After cell-based studies, these compounds can be tested in animal models, and thereafter we can take it to the clinical trials, which is actually the path for our drug discovery. So again, all these compounds showed a very good hydrogen bonding interaction with the target protein, and this way it is not only two but simultaneously using all the right from the genomics to the structural genomics and functional genomics, our group could successfully identify new lead compounds. Again, two important drug targets, both are two important drug targets are therapeutic crucial targets in breast cancer. One is a serine threonine kinase, which is also known as great wall kinase, and the second one is the TET2, which is a transcription factor. Even for TET2, we are now undertaking the experimental work in our group. So see the beauty. We understood the basic gene from ATGC, coding for the different 22 amino acids, folding into a 3-dimensional structure of a protein, determining their structure from either experimental methods or through in silico methods, then understanding their functional relevance in the normal human system, their functional relevance in context of the disease. In our case, our group is majorly working in the breast cancer, breast cancer studies, how different proteins which when get dysregulated are involved in cancer, and from that we could identify initially using in silico approaches the inhibitors that could actually stop the overexpression of these proteins, and these inhibitors as leads now we are validating in our cell-based studies, and we are really getting very interesting results. Maybe down the line, once I have a very good data for functional genomics from NGS, I will be able to show maybe for the next time. So this is the basic knowledge of a gene structure, the basic knowledge of a protein structure actually can guide you to a structure-based drug design. This is how it takes, this is how the flow goes to a structure-based drug design. So with that, I have summarized my work, and I hope the students would have understood something out of it. I sincerely thank again my director, sir, Dr. Anup Anvikar, sir, for all the support that he provides to us for undertaking our research work. And further, I take this opportunity to thank my research team. The lab I am heading, I have a scientist over there, Mr. Brij Bhushan. Most importantly, I thank my student Rajesh. He has been very actively involved in both the works. Presently, he is pursuing his PhD from Italy. Then we have Ashrat, Mahima, Sandhya, Anisha, Chandran, and Varun in our lab, who all are contributing in their own ways to all the scientific efforts that we are taking. So with this, I conclude my talk. And now again, thank you, Dr. Pushpen, and the session I think you can open for the any queries, question, answers, please.
Wonderful. Thank you very much for this very enlightening and very thorough talk. Actually, it actually is very interesting to see that in just one talk you were able to bridge connections between seemingly different aspects of genomics or the molecular biology, from structure to drug designing, and again to its function, and then mutating and confirming that the function is lost. So this was actually very insightful, and I really admire the work that you have carried out, and even more importantly, the way you have explained it to the wide audience here, this was wonderful, in my opinion. I have some questions also from my side, but I would first let the participants ask the questions. So if anybody has any question, they can write in the chat box. And in the meanwhile, when you are writing your questions, yes, there are also some comments from Gauri Mishra that great explanation, madam. First time I have understood the genomics. So that's
Thank you, Divakar. Thanks a lot, Divakar.
And then there are some other questions also. So I'm curious about one of the points that you mentioned about G19B mutation that you carried out. So how you actually zeroed in, or how you chose that this particular glycine you want to mutate?
Okay, so actually when we, as I told you, there were two aspects. We did the structure for this wild type protein. However, we could not get the crystal structure for the mutant. So what we do, there is a in silico method where we actually analyze the functional relevance of each amino acid in that protein. Okay, we actually mutated one by one using in silico approaches all the amino acid residues in that protein.
Okay.
And what we started with the amino acids, especially that were forming the active side of that protein. So actually initially when we did the mutation of all the active side residues, we could actually determine what were the delta G changes, thermodynamic changes, and this glycine 19 valine change was actually contributing to the maximum thermodynamic change, delta G change. So we thought to start with this. So it was an in silico work, in silico analysis that we did, beginning with the active site residue mutation and understanding the free energy change involved with this mutation.
So again, that's very good explanation. Basically, so it was an informed choice by first identifying which one would have a greater impact. So how you determine the flexibility?
Yeah. Pardon?
How you determine the flexibility of that loop?
Flexibility for determining the flexibility, we did the molecular dynamics simulations.
Okay. So
see, we got an insight from that. Glycine at 19 valine was making the maximum delta G change. Once we understood, we went to the bench. We did the experimental work and we generated the mutant. After mutation, actually we did the three-dimensional modeling also. We did the functional activity also, and also we did the molecular dynamics simulation, and those simulations were able to reveal, okay, this is actually reducing the flexibility.
Okay. Wonderful. There are some questions regarding any in silico tool for identifying anti-cancer genes in genome sequence.
अरे ऐसे सामने को पैर हाथ किया रहा है तो देख रहे हो?
Hello.
Keep yourself muted while you are
रहा है ना पागल?
speaking.
I actually somehow I have lost the track. I can read from Swagtam Mukherjee. Yes, ma'am. Thank you. Can you? I can see on chat box from Abhishek. Can you please explain about the process of molecular decking, maybe in the form of a lecture if possible? Sure, Abhishek. Next time we can trouble Dr. Pushpendra. In fact, if you will go to some of my slides, I have actually explained the docking procedure. But in case if you would like, we can always go to the molecular docking. We can take a presentation entirely on molecular docking as well. Sometimes.
So just because there is some meeting all of
Hi, Leonardo. Thank you so much. I am so happy that you are online listening to my presentation. Okay, so you have asked, besides in silico data, have you ever tried another approaches in order to screen new drugs? Yes, Leonardo, I have actually tried the cell-based approaches. I have screened some natural compounds based on literature, and from knowledge-based work, I have actually tried to test them in cell-based studies.
So wonderful. I think I don't see any other questions now. So if there is any other question, you can still write. And I just want
Sir,
to
can I ask?
yes,
in the-
yes, please.
I actually wanted to ask about
Please.
how we can I mean, we were talking about selecting a drug target as a protein. I was thinking about how can we select a potential drug, and we can try to dock it to different other proteins to look for its side effects or how it may interact with other targets that other than what we need. So it could help. Determine the specific side effects of a commercialized drug or existing drug. Is it possible to do so?
Okay, yeah. So now there are holding the nose directly or going around and holding the nose. The question is the same thing. So see, try to understand. There are two things. You know a drug target. You screen different databases to find an inhibitor for that drug target. That is what I have explained in my examples. Now you want to know that you have a commercial drug and you want to see how this drug behaves against different targets, if I'm correct. Is that your question is?
Yeah, this is what I wanted to say.
Yeah.
I mean,
Okay.
if a specific, yeah, sure, go ahead.
Yes.
Sorry.
In that scenario, see, if you have a drug and which is a commercial drug, obviously the data for the commercial drug, many of the things you can get from the knowledge base. You have to do a thorough study. Now, once you know in under what conditions, what is the chemical structure of that drug, against which disease it is known, in that scenario, once you choose a drug and once you have a thorough knowledge of that drug, you can dock that particular drug against different targets, against different proteins. Then you can make a table. The binding energy of this particular drug will be different for different drug targets, right?
Yeah.
From that binding energy, you can have an understanding. That is simple docking studies. You can have a fair understanding. Okay, fine. It is binding to X drug target with this energy, to Y drug target with this energy. Okay.
Yeah.
Now, coming to your other part, toxicity. See, if you have this is a third step. From in silico studies, there are, you can Google it at a later point, ADMET studies. Absorption, distribution,
ADMET.
all that, So there are in silico softwares where actually when you provide an input as a chemical compound, any compound, be it a commercial drug or anything, they will actually give the ADMET prediction, but that will be a prediction. So once you know from the ADMET prediction, you can understand what is the toxicity profile of this particular compound. For as far as commercial compound is there, it's all toxicity studies in animal models, and in silico toxicity can be determined by ADMET analysis. For otherwise, to establish the toxicity, you have to go for the animal toxicity studies in animal-based systems, right? So from you can do the either way. For commercial compounds, everything will be published, so you can have already in advance. You have a fair knowledge of the toxicity.
Okay. I mean, isn't it better to do the in silico toxicity studies rather than going for an animal model and trying to?
If you go for animal model, forget about the animal model. In silico studies are always a chosen thing at the first step. Reason being that because at the first step it reduces your time. You go to the wet lab, you do all the experimental work, and you don't get any response. Means all your time, resources, and precious resources are wasted. But ADMET in silico analysis is always a wiser approach as the first step. But at any point you have to do the validation. You will have an initial idea. Based on that initial idea, you can validate in the experimental work.
Thank you, ma'am. Thank you, ma'am.
I think Swagatama Mukherjee had this question. For protein sequences, we can obtain them from coding region of the gene. However, various carcinomas make us of long non-coding RNAs to aid in disease progression. Is there any way to use these long non-coding RNAs as target molecules apart from protein sequences? See, as far as large non-coding RNAs are there, protein sequences are very like you have different approaches, in silico approaches to model them. Large non-coding RNAs, first of all, you have to do the secondary structure prediction, whether they are forming any kind of secondary structures. So yes, you can do them, but it is really difficult as compared to the protein sequences, because you have n number of bioinformatic methods to model the protein sequences. However, for long non-coding RNAs, first it is always wise approach to establish their functional relevance using experimental methods. To model them and screen for the target molecules, it will be a slightly tricky thing at this stage. Hello. Hello.
Can I write your email also in the chat box, Gauri, so that
the official email gauri.misra@nib.gov.in. It is also available in the directory of the NIB Noida.
Okay. Okay, so I have the Gmail one ready. If I can write that and people can get from you, the official email ID also, is that okay? Or you prefer the official email ID only?
Yes, I will prefer that. It is better to share the official email ID, please.
Okay, I will send that. So I think there is one poll also shared with the participants. That was a question I also asked earlier, and it is related to one exome sequencing session. So earlier also it was asked about how much percentage of the human genome is represented in exome, and at that time also people had answered, 75% of them had answered it correctly, like in the KBC. So wow. 1.5% is the right answer. But there are still some people who think that 10% of the human genome is represented in exome, and some think that 5% of human genome is represented in exome. But it's the correct answer is roughly 1.5% of the genome. There were some other.
So majority is going to be 1.5%, I think.
Yes, roughly 3/4 of the participants. Okay, so this is a repeat question, but in the next session, we would have some more MCQ related to different talks that were organized in this entire past 1 month. So with that, I think it's getting late also on this Saturday evening that you all shared your time with us for this excellent session. And I thank the speaker and all the participants for their time and for their interest. So the next session would be the last one of this series, and you would be informed about it next week. And with that, I thank all of you, and especially the speaker of today, and as well as all the team that has helped in organizing this session, including those who create the flyer, those who create the links, and those who circulate it. Thank you very much, one and all, and have a nice time. Stay safe and enjoy the.
Thank you, Dr. Pushpin. Thanks for this opportunity. I really wholeheartedly thanks all the students. You are always welcome to contact us for any of the queries, wonderful queries you can have. And thank you so much for giving your time Saturday evening. Enjoy your weekend. Have a nice reading, enlightening talks. So thank you everyone.
Thank you. And with that, there would be next month, there would be some hands-on type of training regarding the bioinformatic data analysis of the next generation sequencing data, because in all these talks you have heard about different type of NGS experiments, but it would be useful for many of the participants to understand what type of file formats are there, what is the FASTQ value or the what are the tools that are used for visualizing the NGS data, how to make sense of those millions of the reads. So, but that would be requiring some hands-on exercises, and we will plan it once a month only, because that would require a slightly longer involvement from the team members also. So with that, I thank you all, and please stay tuned. We can share the information through the WhatsApp group that has been created, and through the email that has been available through the registration links. Thank you very much, and have a nice evening. Bye.
Thank you. Thank you.